The last thing that we're going to look at in this chapter is enzymes, and enzymes fall under the big umbrella of proteins. And proteins do all kinds of different things, so we're just going to focus in on one common use of proteins, and that is enzymes. So all enzymes are proteins, and that means everything we've learned about proteins so far is relevant here. So they are proteins. We could also say that enzymes are biological catalysts. And catalysts, if you remember, we're back in chapter 5 if you want to review that. Catalysts help speed up reactions by lowering the activation energy. So check back to chapter 5 for that. And we're going to look at a couple different pictures to help us see and understand enzymes here. All right, so I'm going to actually draw a really simple picture for you first, and then use these more complex ones to understand a little bit better. So with an enzyme, we can understand enzymes with 3 pictures basically. So this would be my enzyme, and remember, an enzyme is a protein, so this wouldn't really look like Pac-Man. It would be full of ribbons and curls and beta sheets and stuff. So this would be an enzyme, and E for enzyme. And I would call this region right here the active site. That's the region of the enzyme where the substrate, S for substrate, is going to come in and fit right in. And I'm going to color my substrates green on this page. All right. So I have an enzyme, which is a catalyst. I can use it over and over again. So let's add that here. Enzymes are reusable also, like all catalysts are. The substrate fits right into that active site, and so what happens next is we get what's called the enzyme substrate complex. And this is where the enzyme is actually helping something happen to that substrate. It might be helping break a bond, it might be helping form a bond, but that substrate is fitting in nicely with that enzyme. And the reason it's fitting there is that there are going to be some intermolecular forces usually, maybe a temporary bond or two also, but holding it securely into that active site. And then after that, the enzyme has helped the substrate change. And so we're going to release, maybe we were trying to break it, release that substrate. Now it's 2 different products. Here is my enzyme, empty and ready to cycle back and be reused again. And my products can go off and find another enzyme or be used in some way. So we have our enzyme with substrate, enzyme substrate complex, and then my enzyme that's done and it's ready to go back up here. If we can look at a zoom in of this active site area up here, this is also showing you an enzyme. So this would be my big enzyme here. And then here's my active site, and all of those little knobs sticking off, those would be side chains. So all these little circles here are side chains. And so those are going to reach out and form attractions or even sometimes bonds, but it's generally going to be just attractions between the substrate and the enzyme. And so this is just kind of a zoom in where you can see more of the chemical features of this kind of attraction here. Along with that, if we want to get a little bit more detailed about our enzymes and our substrates, here's another picture where you can see there's more going on in the active site. All right, so the enzyme is going to be this middle piece here. Substrate, here I actually have 2 substrates. So we have this dark substrate throughout, substrate 1, and then we have substrate 2. And the enzyme is going to make the reaction easier by holding them both securely in place. And then there's one more new piece in this, and that's going to be, I'm going to use pink here for my coenzyme. And so a coenzyme falls under a bigger umbrella of molecules that are called cofactors. So a cofactor is either going to be a metal. This is why we consume a lot of traces of metals, is to help with reactions like this, or something organic. Some small organic molecule, and those small organic molecules are called coenzymes. We've seen some of these, for example, FAD and NAD+ are coenzymes that help with reactions, and these cofactors help our enzyme do its job, and they are also reusable. So an analogy that I think of sometimes, here's that cofactor. So the cofactor, I think we're going to skip the analogy, but you can see the cofactor sitting here, and it helps maybe grab a phosphate from one substrate, and then the cofactor rotates and moves that phosphate to the other substrate. And so now that phosphate is on product 2 and product 1 has lost it. So the enzyme and the cofactor and the substrates all work together to generate those products. And then I can take the enzyme and the cofactor or coenzyme and bring it back up here and recycle them over again. So that is a good example of a enzyme kind of in action with a little bit of help from its coenzyme. Next up, we'll look at some inhibition in a second, but I guess before we do that, one more place we can see this. So this would be a glimpse. This is one subunit of a lactase enzyme. There's actually 4 subunits in lactase. The enzyme is huge, and maybe somewhere in the middle here, this is not necessarily the correct spot, but that's where the lactose would come and actually get broken in half. So enzymes are relatively huge compared to the substrates, and that is kind of how we can connect it back to these big pictures that we saw at first. So next up, we will look at some ways to inhibit or stop this enzyme activity.